Well, Dr. Gilbert, I can't tell you what a thrill it is for me to be sitting with you. I was a Navy medical officer in San Diego in the mid-'80s, and I read Invisible Frontiers and was inspired by the story and devoted my life to diabetes and better treatments for it. And Invisible Frontiers made a huge impact on me.
Oh, that's delightful. It's a wonderful book. I thought it was actually a very vivid description of both the search for the insulin gene and the effort to make recombinant insulin, and the development of the companies, the growth of the origins of Biogen and the Genentech. There is that wonderfully vivid description in the book of our entire trip to England to try to clone the human insulin gene.
Right.
We have a wonderful trip, and we work very hard, and we drink a lot of wine, and in the evenings as we get out of the lab. And we thought we were successful in bringing the samples back to Harvard. We discovered that we've recloned the rat insulin gene because there had been a contaminant on the glass plates and the glassware.
Just reflect for us the impact of that era on modern medicine.
Is the development of the biotechnology industry. And in fact, the entire effort to make first useful human genes in bacteria or in other organisms. That's produced a wave of pharmaceuticals that have had a great impact on people. The entire development of antibodies as made then in cells and then used as drugs are still a dominant pattern of drugs today. Small interfering RNA molecules have become drugs. The drawback with the protein drugs, just like the drawback with insulin, was that it was something that you had to inject. And you couldn't take it by mouth, and so that the question of whether you can have an oral drug is still a dominant one. There's still an effort, I think, with insulin to get it into the body in other forms.
Yes, sir. What can you reflect any on Jeremy Rifkin and the impact that had on your work and the political atmosphere?
Is there conceptual possible difficulties with this idea of cloning genes and moving pieces of organisms around?
Right.
And so I think it's actually in the Gordon government since '74 that we write a letter to NIH saying we should look at the government should think about, is this a problem?
Right.
And shortly after that, we actually get federal guidelines as to how to do the recombinant DNA research. And so one line of scientists is thinking, yes, we're using safe bacteria, we're taking cautions in how we do the research, we're using special laboratories if we need them, but one side of the scientific community at Harvard began to argue against it. And that argument first was in front of the dean, a curious moment in which Ruth Wald talks about how unknown and dangerous the science is, and you shouldn't explore the unknown. And the dean says, but I thought the point of science was to explore the unknown.
What would you like your legacy in all of this effort to be?
Well, my legacy is actually in the DNA sequencing, and so it's which is of course now bypassed all of that.
Yeah, right.
The fun part of science is that everything we do gets transformed, and if it's successful, the world moves out far beyond it. Fred Sanger in England and I in the States found ways of working out DNA sequence very easily and very rapidly. And as I said, the problem before that was one of considerable difficulty. We could work out one base a month with great effort. But in any case, we had worked out that first piece of sequence. Then we discover very rapid methods. And those methods get used, we give them away. And they begin to get used around the world in '76. By '86, about 8 million bases of DNA have been sequenced everywhere in the world. By '96, it's 800 to a billion. And by 2000, it's already the human genome of 3 billion. The process went very, very rapidly. The chemical method, which I and Alan Max have developed, was used for about 10 years, and then as the machines came in, Fred's method began to be used entirely, and then, but by now the machines have gone far beyond that.
Yeah.
I mean, the human genome, first human genome took about, in actuality, about $600 million to sequence, and 5 to 7 years in many laboratories, and the machines now will do a human genome in about 30 minutes. And the cost is now somewhere probably about $200 to $300.
It's amazing.
So it's a total, wild transformation, and new machines are coming in constantly.
You are quoted as saying, more or less, it takes as much effort to answer a small question as a big one. So then don't waste your time on small questions.
Yes, I do say things like that.
Yeah. But I wonder if there's room for that in science anymore.
Well, it is. There is in places.
Yes.
It is one of the problems that so much of the science has become big science. It is then hard for people to function
right.
in the way which we could. So when I began working in messenger RNA, when I started biology, I worked on messenger RNA, which is a fundamental discovery. You worked on ribosomes, we worked on mechanisms of protein synthesis, discovered certain interesting facts about how you made proteins. And then I went off and worked on the repressor. And each of these has become gigantic fields of
yes.
science and
yeah.
full of fantastic detail. But the amazing thing is, people make totally novel new discoveries in science all the time. And when those fields are new and novel, then they're simple and they're easy to do, and
yeah.
you can do something interesting.
Yeah.
We think of humans as having 20,000 to 30,000 protein genes, plus another many thousand small RNA genes and other things floating around, and all sorts of RNA molecules floating around, most of which are of unknown function. And each of these genes is modified post-translationally in many, many ways, so that all of those modified by alternative splicing, modified by chemical modification. Complicated story, fantastically complicated.
Yeah.
And it's surprising that people can find totally new things
yeah.
in that, but they do.
Yeah.
So there are totally new things coming out in science.
The Levine Symposium is well attended by junior scientists. Any reflections for them? Any comments to them that you'd like to share?
Well, I think science is some of the greatest fun you can have, is
yeah.
doing science. As scientists, we're constantly interested in the latest thing.
Yeah.
Sometimes it's useful to know a little past history, past knowledge. I think the best advice is the one you just quoted to me, which is actually find good projects, work on things that have significant outcomes.
That's great. This is a real thrill for me.
Thank you. I constantly take photographs everywhere I go. These years I use the iPhone. Before that, I used to carry, that's 15 years ago, I'd carry a small digital camera in my pocket. I used the camera all my life, going back to a film camera when I was a child, and, home developing, darkroom, enlargement, all of that. And then as the digital, then in the later years, I would just use the camera to record things that I traveled, or to record instances of antiquities and things like that. But in about 2002, I discovered I could make very large images from a small digital camera, and began to say, well, images on this 2 by 3 foot scale then begin to have an emotional impact. And so I began doing the photography as an art form. So these images, something like this, intensely colorful, use of black to outline things. It starts as a photograph, and then several copies of that photograph are superimposed to make an image. It's actually based on a building in San Francisco that has these striking fan lights in it. And so the building looks a bit like that, but not quite like that. And then I made it's a 5 foot high image. This is an image that I call Doors to Nowhere. And it's actually taken in a museum in Rome. And it is a set of several doors, but they've been superimposed on each other to make this extensive image. And I play with the image in the computer. I actually drive the colors to 100% saturation. And that produces sometimes these strange and wonderful textures that I play with. The most recent images are actually very striking images of flowers and plants. And they're images like this. This is actually a plant from our garden, which doesn't look at all like that, of course. It actually is a rather white prism plant. And that's a sort of composite, strange composite image.
Art rays.
Oh, that's lovely.
Peter, you know Peter Medico.
Oh, yeah.
You're the hero of that book. That's terrific.
Yeah, it's really, I thought it was a wonderful book. It sort of has a 6-week life.